Hello, thank you for joining us for our webinar. This month, I'm delighted to have Dr. Kate Kaczurczyk. Kate is a principal research scientist with Battelle Memorial Institute in Columbus, Ohio. Since joining Battelle in 2014, Kate has served as the principal investigator on projects focusing on monitoring and application of bioremediation processes in contaminated sites. Kate has detailed knowledge of molecular biological tools and the assessment of remediation technologies, and she's currently a principal investigator at Battelle's Environment and Infrastructure Business Unit, helping to aid in study design, assay selection, and result interpretation. Today, Kate's talk is going to focus on the new tool that we have with Proteomics, and this tool is something that's new in our toolbox that allows us to better assess remediation and help us to achieve our cleanup goals. Now, as many of you who have attended before know, the webinar today is being recorded, and it will be available on the resource section of the Microbial Insights website within the next week. A link is also being automatically emailed to all registrants as soon as it's available, so you don't have to contact us and ask for this link. It'll automatically be sent to you. At the end of the webinar, there's going to be a question and answer period. So please go ahead and type your questions into that panel from your GoToWebinar software. And at the end of the presentation, Kate will answer as many of those questions as we can. If you have additional questions that you think of after the presentation, you can always email them to us at info@microbe.com, and we'll get with Kate and get those answers for you. So without any further ado, I'm going to go ahead and turn it over to you, Kate.
Thank you so much, Dora. I appreciate this opportunity, and I'm happy to talk to you all about proteomic analyses for microbial communities and in environmental microbiology. This work has been performed at the Battelle Memorial Institute that I work and has been spanning not only the environmental remediation field, but also application of those advanced omics in forensics as well as health. So a brief content of today's talk will start with the introduction to molecular biological tools and really focus on those advanced MBTs, including proteomics, both shotgun or discovery proteomics that helps us to characterize diversity of proteins or those express activities of molecular of microbial cells, as well as targeted quantitative proteomics that helps us to quantitate protein abundance in our samples. And for the introductory purposes, we also talk a little bit about the cost of proteomic tools in comparison to conventional tools and other advanced tools. So why are we really interested in microbiology as environmental practitioners? Microbiology provides insights into bioprocesses that occur in the subsurface, both biogeochemical and biodegradation related. The microbiology helps us to understand what specific microorganisms are present in the subsurface. Are those microorganisms active? And if they are active, what type of specific processes they are focusing on at a given time of sampling. Microbiology also helps us to understand how many different types of microorganisms are present at the site and what specific functions they are playing at the given moment. There are a variety of different molecular tools present in the market nowadays, and all of them are rooted in molecular biology concepts within which we study the gene structure and function at the molecular level. The field of molecular biology also overlaps other areas such as biochemistry, in which we learn a lot about structure, function, and interactions of macromolecules, and genetics that studies presence of genes in intersequences. So a variety of traditional conventional molecular tools are available in the market, and you may be familiar with qPCR or microarrays. These may not provide sufficient information to assist in site assessment and remediation. However, more advanced omic tools are starting to be phased into the environmental market nowadays. And these help us to understand complexity and function of microbial populations in the subsurface. So if we take a deeper look into the bacterial cell and its composition, we know that every microbe needs nutrients, electron acceptor, and electron donor to generate energy and convert carbon-containing compounds. There are a variety of different molecular structures present in microbial cells that help the microbes to grow and convert substrates of a variety of different origins. DNA is our primary information storehouse. It tells us what specific metabolic genes are present in the specific DNA sequence, and also inform on identity of specific microorganisms in the environment. DNA is later on or next expressed into mRNA, which is our messenger RNA molecule. It takes information from RNA to ribosomes in which proteins are being produced. So messenger RNA has a capacity to tell us whether our microbes are active in terms of expression of genes into messenger RNA products, and if genes are being made. So these two specific areas of science are backed up by molecular tools such as quantitative PCR or microarrays, as well as metagenomic sequencing, in which information from DNA is being extracted and provided to us practitioners. In terms of mRNA, we do have metatranscriptomic tools that help us to quantify the concentration of transcripts in microbial cells. In terms of proteins that are produced in ribosomes, metaproteomics or proteomics helps us to understand proteome diversity, complexity, and helps us also to quantify protein abundances in microbial cells. So all of those specific types of molecular tools somehow correlate to each other, with genomics informing us on DNA sequencing results, gene abundance, and identities of our microorganisms, transcriptomics, helping us to understand what type of transcripts are being produced in microbial cells, and proteomics, which is basically a science of proteins. It helps us to understand complexity and abundance of proteins in our samples. So all of these three categories of different tools fall under functional genomics, which is a field of molecular biology that uses data from all of those omic tools to describe function and interaction of microbial communities. There is one more tool that is recently getting a lot more traction, called metabolomics. Metabolomics is basically a science of small molecules that are being produced by microorganisms, and then can inform on specific pathways and processes that are ongoing in the cell. The conventional and advanced molecular tools are just an amazing array of helpful techniques that can assist in answering a variety of different questions for us environmental remediation practitioners. They can determine if remediation is working. They can help us to select an appropriate remediation strategy. They can help us to transition from active treatment to monitor natural attenuation. And they also can be included as additional line of evidence in assessment of natural attenuation. However, to be able to correctly apply any given molecular tool, we really need to think of a strategy that includes a variety of different questions, starting with what our project goals are, moving into what phase of remediation is our site in, and what our geochemical data is saying to us, in terms of spatial and temporal variability of the ecosystem, and also what other lines of evidence are telling us. So all of those questions really need to be considered very carefully before we move into applying any given tool. And understanding what is our objective is paramount to correct application of these tools. So if we look deeper into a DNA-based molecular biological tools, these tools provide us with information on potential for quantitative degradation rate information. qPCR is telling us about the gene abundance. Metagenomics helps us to understand total microbial community composition, as well as relative gene or organism abundance in a cell, in a sample. However, these tools, again, provide only information on potential for that specific activity. Now, if we move into proteomics, proteomics informs us on functional activity and this actual activity in microbial cells. There are two different types of proteomic approaches that I will be talking about in greater detail today and giving you some case study examples, starting with shotgun proteomics. So shotgun proteomics is this really interesting technique, an ideal technique for identification of proteins in post-translational modifications. This technique is semi-quantitative and requires metagenomic sequence data to be provided. Why? Well, there are so many different microorganisms in the environment, and most of them can express similar activities. But sequence of proteins of a specific activity related to one microorganism may be different than that same sequence related to a different microbe. That's why we do need to understand the composition of microbes that are in our samples so that we can derive a well-educated database of proteins that can be linked back into those activities. So shotgun proteomics is really a global proteome mapping. It can help us to understand expression of proteins among different samples at different sites, but can also help us to find new and very unique biomarkers of specific processes that we're interested in. Now, quantitative proteomics or targeted proteomics, as you will probably hear me say today, this is a protein quantification in which we really need to know and have a predefined protein target so that we can quantify their abundance in the sample. So application of the omic technology, since we're talking about proteogenomic approaches here, in which we're utilizing information from genome sequencing that provide basis for protein annotations and protein searches in our samples with proteomics have been a focal interest of the groups that I work in at Battelle. So far, we have been studying several different types of contaminants that proteomics is helping understand their conversion, starting with MTBE, chlorinated volatile organic contaminants that a lot of you probably work with, munition constituents such as RDX and TNT. We've also worked with petroleum hydrocarbons, PCBs, and we're involved in several projects that try to understand microbial-induced corrosion through the prism of metagenomic and proteomic data analysis. So moving through discovery proteomic pipeline and understanding step-by-step how this process works, I prepared several graphs so that you can visualize the process. So for this specific tool, we can either start with a single pure culture that is grown on specific substrate, and we can observe differential expression of specific proteins over time, or we can look into samples that are taken from a contaminated site and try to understand whether our contaminant of interest is being actively degraded by a given microbial consortium. So either way, you will always start with a grab sample from either pure culture or microbial consortium, and this sample will be subject to sample processing. This process is uniform for both discovery proteomics and for targeted proteomic platforms, and entails lysis of microbial cells, protein extraction, reduction and alkylation of our proteins into shorter pieces called peptides, and then cleanup of the sample to make sure that we remove all of the detergents and salts, and that sample can be injected into LC-MS coupled with quadruple time of flight. So this is a very specialized piece of equipment that helps us to acquire data on mass and charge ratios of specific peptide precursors in our cell, and then link them back into a database in which we identify peptide hits resulted from the specific sample analyses. So having this in hand, we can build additional methods for accurate and sensitive sample processing. I wanted to give you a little bit more tangible data and examples so you can understand how this really works in practice. So we use discovery proteomics as one of the lines of evidence to investigate natural attenuation of MTBE at a contaminated site. This project was funded by the ESCCP, and we were using both metagenomics and metaproteomics to understand microbial community composition at the site, looking for potential for that activity. And we're applying discovery proteomics to detect specific proteins of MTBE degradation. And that helped us to evaluate whether degradation biomarkers of MTBE are present and hence inform that degradation activity. So there were two sites that we looked into. First, the 22 area gas station. This site has been located in California and had been contaminated with MTBE. The active treatment part of the remediation at the site was discontinued in 2012. And we were looking into reduced MTBE concentrations over 2 orders of magnitude over time. The remedy that was selected for the site was a 2-tier biobarrier system and oxygen sparging. So after the treatment was discontinued, the site was progressed into a long-term monitoring, and we were observing very low MTBE concentrations at several wells, lower than 10 micrograms per liter, and mostly anaerobic conditions. We performed the M&A assessment at the site, looking into a variety of different lines of evidence, starting with concentrations of MTBE, as well as its byproducts of its degradation, such as TBA. We looked into geochemistry conditions at the site and implemented those 2 omic approaches into investigation. Now, we also used a control site, 13 area gas station, which was located approximately 2 miles away from 22 area gas station. An active treatment of MTBE is ongoing at the site. MTBE and TBA are both detected, which signifies that the applied remedy is successful. And we were expecting to see both metagenomic profiles of microbial communities and metaproteomic profiles with presence of MTBE degradation proteins at this site. So MTBE can be degraded with 2 different processes, both direct mineralization of MTBE is governed by microorganisms such as Methylobium petrolestium PM1, which is our indicator species for qPCR assays, and cometabolic degradation of MTBE that happens in presence of other substrates like pentane or butane And alkanes in general. And MTBE here is not fully degraded to carbon dioxide, but produces some additional bottleneck products that are later on slowly degraded over time. Proteins of MTBE degradation of this direct metabolism are listed on a figure presented in this slide. Starting with MTBE monooxygenase, moving through TBF carboxyl esterase or HEBA mutase. All of these proteins are involved in full conversion of MTBE to carbon dioxide. And we were looking for those proteins in all of our samples that were analyzed in the study. So if you look at that same graph of our site, we saw that there were no conclusive data in terms of degradation of MTBE and trends of MTBE degradation over time. We found that trends of MTBE degradation were either stable or non-decreasing. Our geochemistry didn't really give us enough information on the site conditions, and most of the times what we were seeing were anaerobic trends for oxygen concentration. But when we really looked deeper into metagenomic composition of microbial communities at the wells that we selected for this analysis, we saw a vast diversity of microorganisms that were related into aerobic direct and aerobic cometabolic degradation of MTBE. We also found few anaerobic cometabolic MTBE degraders. When we searched for proteins related into MTBE degradation at the site, we did not find any of that specific pathway that I just showed you. But we found a lot of proteins expressed by microorganisms that can degrade MTBE. And this means that the MTBE degrading microorganisms are active, and they're still growing at the site even after the remedy was phased out. This also signifies that the site has the potential for MTBE degradation, but maybe concentrations of MTBE are too low so that we can see any expression of those specific proteins. Or maybe the biomass of those specific MTBE degraders is too low for us to detect it with, to detect the proteins with methods on hand. Now, when we looked into degradation of MTBE at 13 area gas station, we noticed that we were definitely seeing aerobic direct MTBE degraders, as well as aerobic cometabolic degraders present at the site, especially in the source zone at the site. And we also saw presence of proteins of direct and cometabolic MTBE degraders and presence of proteins of MTBE degradation pathway. And that was the direct proof that MTBE degradation is happening at 13 area gas station. So if you look back into that MTBE direct mineralization pathway, you see that out of at least 10 different proteins that are involved in this degradation, we were able to detect at least 6 of them, which is a very strong line of evidence for an active degradation of this contaminant. Another case study that we were involved in was using both of these tools, so metagenomic sequencing and proteomics, to find linkages in microbial composition and protein content that affected microbially induced corrosion. And we were asked to analyze samples from the seawater injection system that was prone to microbially induced corrosion, and wanted to investigate what can we do to help our clients to minimize those microbially induced corrosion effects. So again, 16S was used to characterize microbial community composition, and shotgun proteomics was used to understand what specific proteins that are related to the MIC are present in our samples. We collected samples from several different elements of that seawater injection system starting with the raw unfiltered seawater, moving through the pipeline downstream of a treatment plant, and collecting samples of pig envelopes, which are those very slimy and biomass-containing samples, and analyze them with both whole genome sequencing, 16S sequencing, and proteomics. So the interesting thing that we found here was that all of the samples and their microbial communities were dominated by one very specific microorganism called Sedimentacola stellanati reducens. This microorganism can convert perchlorate into chloride ions. An active chlorinator was present at the site for disinfection purposes. And this species also can convert sulfur and nitrogen oxides. And conversion of those two specific substrates is directly linked into microbially induced corrosion. So we looked closer into functional genes and presence of enzymes that were expressed by this microbial community at the site. And we found that a variety of different sulfur oxide reduction proteins were present, starting with the sulfite reductase, thiosulfite reductase, adenylyl sulfate reductases. All of these were actively expressed and present in the community of microbes that we found at those different injection points. So this is a pathway of sulfur oxide reduction, and you can see that majority of the peptides that we found linked back into proteins involved in sulfur oxide reduction. When we moved into nitrogen oxide reduction process, we also found presence of functional genes and presence of their transcripts. So their transcripts and proteins. Nitrate reductases and nitric oxide reductases were present in fairly high concentrations, and we detected high abundance of hits per million in our whole genome sequencing data. So again, this is a pathway for nitrogen oxide reduction, and majority of the proteins that we found tag back into this pathway. So with all of this information, we looked back into proteomic data, and we tried to understand where those proteins come from, and most of them were identified and mapped back into Sedimentacola stellanati reducens. So having this knowledge, we were able to help our client to design a specific biocide system that was injected into our pipeline and basically helped to wipe out the specific microorganisms that contributed to microbially induced corrosion. So this is a really good example of how integrated omic analyses can be helpful to our practitioners in real life. And with Discovery Proteomics, we have this very powerful tool that can help us to enhance site characterization, optimize remedy selection. It helps us to enable remedy modification, either from passive or active, or switch in a biosite that will target very specific microorganism that is contributing to adverse effects in our system. Discovery proteomics can also help us to determine effectiveness of existing remedies to expedite site closure and ultimately provide time and cost savings. So fairly attractive tool. It's still new for the environmental practitioners, but I think it has a pretty good future application. Now, when we talk about the environmental remediation, understanding of biodegradation rate is very critical for our site management. And here we can measure biodegradation rates in our field samples, but it's a quite difficult process. If we have limited data in terms of sample volume, frequency, replicates that we have in hand, a lot of times we don't get as many samples as we want to provide a very robust and efficient characterization of our site. And also challenging plume dynamics can be fairly can be affecting our success of measuring the biodegradation rate. And we believe that proteomics can bring benefits to this process. It can provide lines of evidence to further support decision making and eliminate requirements for performing extensive sampling at sites. So targeted proteomics has a very specific application here. Again, the pipeline or the process in which samples are analyzed with this tool is similar to the discovery proteomics that I already discussed. And here it is paramount that we have this microbial community metagenome information, and we provide an upfront identification of very specific genes of interest and annotate them into proteins, and use these as a very targeted approach to quantify biomarkers of the degradation of a specific process. So after samples are collected, proteins are again extracted, reduced, alkylated, and digested into peptides. And peptides are then analyzed using triple quadruple mass spectrometer, in which we're looking for a very specific mass to charge ratio of those peptide fragments for quantification. What follows right after is establishment of method detection limit for selective peptides. And here we're using isotopically labeled peptide equivalents so that we can provide this quantitative estimation of abundance of our proteins in the sample. So once this is established, we also like to know what is the lower and upper limit of our quantification as well as detection, so that we establish our linear dynamic range for peptide concentrations, and are able to accurately and precisely monitor concentrations of specific biomarkers in that sample. And this is important from the sensitivity standpoint. So this is another case study in which we are looking into degradation of chlorinated solvents and implementing an integrated approach in which qPCR, transcriptomics, and metagenomics and metaproteomics are included. For chlorinated solvents chemistry, we know that PCE is degraded into TCE, cis-DC, and vinyl chloride, and a variety of different genes and proteins are included in this specific process. The reductive dechlorination happens with the use of a reductive dehalogenases, so that's how we call our proteins. And there is again variety of different flavors of them. For example, TCEA or VVCA that are produced by Decalococcoides or the Sulfobacterium decalobacter listed up on this graph. So the knowledge of those specific target peptides or target genes is very helpful. The metagenomic sequencing that allowed us to identify those very specific biomarkers was run for one of the microbial consortia that we use in this study. SDC9, this is a consortium that Aptin provided to us, was sequenced using 16S sequencing and whole genome sequencing, and we're able to identify RDase genes that encode for specific RDase peptides in this community. Those correct RDase peptides then were used to identify peptide annotations in SDC9 culture extracts, and this approach helped us to avoid false negative in relation to using only NCBI or any type of database available approaches. So through shotgun proteomics and metagenomics, we were able to identify at least 14 unique RDH genes and annotate them into proteins. And these specific proteins and peptides that encode back into those proteins were later on used to develop a method for this targeted proteomic approach. We're able to establish instrument detection limits for every single peptide that we're using in this approach. We also determined method detection limit and supernatant of SDC9 culture, and determined which of those peptides are the most sensitive quantifiers for targeted proteomics. Some of these peptides have lower sensitivities than the others, and that's to be expected. Not all of them will be perfect quantifiers. Some of them will be only qualitative controls so that we can understand if the coverage of those peptides is tying back into a very specific protein. So we used samples of sediment and groundwater collected at Joint Base Lewis-McChord. That is our demonstration site. And these samples were later on advanced into a microcosm setting in which was amended with SDC9 microbial cells at different starting concentrations. And what we wanted to understand here, and this study is still ongoing, so not all of the data is available, we wanted to understand whether we can tie together kinetic rates of degradation of chlorinated solvents to biomarkers of their degradation. And these included genes, transcripts, and peptides. So we wanted to obtain first order degradation rate coefficients and then correlate them into the peptide abundances in the microcosms. So some of the preliminary data shown here, we were able to derive the kinetic rates of degradation of cis-DC vinyl chloride, and then couple them with the abundance of specific RDA peptides. So this is the top graph on this slide is showing our first order rate degradation of cis-DCE that was spiked into our microcosms, and then your x-axis is showing your density of peptides in femtomoles per milliliter. And these tie back fairly nicely to the rate constants and show a fairly good correlation of these two variables. So for targeted proteomics here for this study, our main goal is to tie the degradation kinetic rates of chlorinated solvents to the abundance of peptides and be able to predict in-field rates of degradation of those chlorinated compounds. Targeted proteomics also allows us to help and optimize remedy selection and quantify abundance of protein biomarkers and correlate them back to rates, which in overall provides time and cost savings to our projects. Now, when we look at the cost comparison of this tool to other molecular tools that are used by practitioners, this table has been adapted from the ITRC and updated a little bit. We're looking into a cost that spans between hundreds to $1,000 per sample cost, depending on the depth of the analyses. Shotgun proteomics is a little bit more expensive due to the fact that we need to obtain some metagenomic data, develop the platform, identify our protein targets, our peptides, and tie them back into the metabolical processes that are occurring at the time of sampling in our microbial community. The targeted metaproteomics is a little bit more affordable, and the data for cost here were derived from a variety of different institutions that are offering those tools, as well as academic labs. And they, again, vary dependent on where this analysis is performed and how well the group of biomarkers is defined and characterized. So why is proteomics? Well, I think that in the age of application of this variety of different omics tools, proteomics can help us to provide rate predictions, give us a more educated information on actual degradation rate of contaminant, as well as provide project cost savings and possibly expedite response and site closures. So this is definitely a tool that is new to the environmental remediation managers and environmental practitioners. The proteomics and the basis of its application are not really that new. They have been present in molecular diagnostics for human health for many, many years, and the principles of sample processing and application of data from those analyses are just recently being slowly phased into the environmental space. So I wanted to thank all of my colleagues and coworkers from both the Battelle team, as well as a variety of different organizations that have been involved in application of proteomics and metagenomics into site assessment, and extend great thank you to all of you for helping us advance the science, and I'll be happy to take questions.
All right, thank you very much, Kate. That was a great presentation. So if you have questions for Kate, please go ahead and type them into the question and answer panel on your GoToWebinar screen, and we'll go through these. So the first question I have, Kate, is talking about MTBE monooxygenase. And in MTBE metabolizing bacteria, we know that this is membrane bound rather than soluble. Does this have any impact on the analysis that you're doing?
No. So we have a variety of protocols that are designed to extract those membrane-bound proteins, and we can fractionate the microbial cell dependent on whether the proteins are present in the cytoplasm or they're membrane-bound. And that can be done, and if we know upfront that our protein of interest is bound to the membrane or membrane bound, then we can use a different protocol to be sure that that specific protein will be extracted. But this is not really that difficult to do, and should not be problematic.
Okay, great. You mentioned several contaminants that you've worked on in your slides. Have you done anything for some of the newer emerging compounds like 1,4-dioxane?
We have not worked with 1,4-dioxane just yet, no.
Okay. All right. In your MTBE case study, can you explain the differences between the X's that were on your chart? Like, how often were you able to detect proteins in the biodegradation pathway versus other proteins that matched organisms thought to degrade MTBE?
Sorry for scrolling back here, but so this is the 22 area gas station. The red crosses show detected direct and cometabolic MTBE degraders, and then the green axes show the presence of proteins of direct and cometabolic MTBE degraders. So because we were not able to find MTBE degradation proteins, but we found signature proteins of those MTBE degrading microorganisms, we know that they are active and they are present at the site. So we were not able to find them here, and we know that there must be specific threshold of contaminant concentration at the site at which the microbial cell starts to produce more and more proteins. We also know that probably there is some detection limitation for us within which we are not able to see those proteins if the proteins expressed in a sample are below. So for 13 area gas station, we were able to find both proteins of MTBE degradation pathway, and they were present mostly in the source zone, close to the area of the contamination, but we're not able to find them in the further distal parts of the plume. The proteins of direct and cometabolic degraders, though, were found across the whole site. So again, we think that there may be a threshold for detection, or maybe there is some specific concentration of MTBE that those microbes need so that they start to degrade it, and it becomes the carbon source for them. So that was one of the limitations that we found during the study.
Okay, what are your sample volume requirements for the proteomic analysis?
For pure cultures, we take as much as 2 to 3 to 5 milliliters of a community. We found that it's not the volume of the sample, but abundance of microorganisms that is important. So with the 10 to the 6th of 10 to the 7th total microbial cells, we have plenty of material for to work with, and it can be million cells per liter, or it can be million cells per milliliter. We have methods available in-house that help us to reduce that sample volume and then work with it. So we take really anything that the client can give us, but we advise that for targeted proteomics, we find that the threshold of 10 to the 6th cells in a sample are necessary to have so that we can see those peptides in through our instrumentation.
Okay. There's a question wanting to clarify when you're talking about quantitative proteomics versus total whole proteomic analysis. So it says with quantitative proteomics, you're focused on specific peptide sequences instead of the whole proteome. Is that correct? And if so, could you please explain the benefit in looking at those specific peptides instead of the proteins? I'm not sure that I understand that part of the presentation correctly.
Okay, so the approach that we're using is it's called a bottom-up proteomics, in which we are resolving peptides. And these peptides link back into a specific protein. And we assume that there is one peptide per protein. So if you see specific peptide, then you can make an assumption that you do see that specific protein. Now, in the same sense for the quantification. So if we see abundance of a peptide, we correlate it back to be the same as abundance of a protein. Does that answer the question?
Yeah, I think so. Okay, now you talked about when you're doing analysis of samples, do you have a preference on looking at soil versus groundwater? And if it's groundwater, do you recommend that there's some type of sonifier to help increase the microorganisms that are present in the groundwater? Have you done any tests to look at those?
That's an interesting question. We don't have a preference. If you like to give us soil, we'll take it. If you would like to give us groundwater, we'll take it too. The extraction procedure from soil is a little bit more elaborate, but we do have protocols developed for that. In terms of groundwater, we've worked with Dora, and we started to look into some of those bioflow filters that you guys developed, and we find them very helpful in terms of filtering water in the field settings, so that you don't have to provide big volumes, but you only provide us the filters. We have not tried sonication, no. We have a couple of methods in mind that could be helpful to concentrate biomass in fields, but these are in early stage of development. So hopefully we can share these soon with you.
Okay. When you do this type of testing, do you recommend any type of metagenomic analysis be performed or done independently to be, used in conjunction with the proteomic analysis?
Yes. we love to have metagenomic data up front, and that helps us to build a database of proteins that are present in your specific microbial community. So if you can imagine looking for proteins in microbial consortia from environmental sites, it's looking like for a needle in a haystack, especially for looking for those very low abundant proteins. So to be more precise and provide a more accurate analysis, we always, or mostly all the time, we prefer to perform metagenomic sequencing of your community so that we know what microbial species are present in that community. And then we derive protein annotations or acquire protein annotations for those specific microorganisms from databases. And our protein searches are then based on microbial variability in that specific sample.
Great. And this is one of the things that Battelle and Microbial Insights are working on together, is how these analyses can complement each other. And then once you do the proteomic analysis and you identify markers that are of interest, there are ways that we can design targeted qPCR assays to help you easily and cost-effectively track and be able to identify and quantify those organisms or genes over time. So that's one of the things that our partnership is helping to provide.
Right, and I think a good example there is this MTBE study where you only have qPCR probes for MTBE monooxygenase, but you could hopefully, derive probes for the other enzymes in that specific pathway from the metaproteomic data that we had derived.
Definitely, and we found that with some of the corrosion work that you did as well, that we can look for markers that we can then use moving forward in the assessment.
Absolutely.
So, very exciting time. Well, thank you very much, Kate. Kate, are you willing to share a PDF of your slides for the attendees today?
Absolutely, I'll send it over to you.
Okay, great. So if you are interested in that, just email info@microbe.com and we'll send you out a PDF of the slides. And again, Kate, thank you so much for your time and going through this topic. I'm excited to see, how this tool evolves and what we're learning and new markers that we can discover through the use of this new technology. Thank you everyone for joining us today and being part of the webinar series, and we hope you'll join us again next month, and I hope everyone has a wonderful day. Thanks again, Kate.
Thank you. Have a good day. Bye-bye.
Thank you.